So hey everyone, welcome to Core Vet. So today in this particular video lecture we'll be discussing about anthrax. Okay, it is quite an important disease whenever we are going to enlist quite an important bacterial disease in the case of the veterinary field. Then anthrax holds quite an important position in that list. Okay, so starting with the synonym names of the anthrax. So the very first name is the splenic fever. Then it's the Mills brand, which is famous in the case of the Germany. Then the next is the Chavan, which is famous in the French. Okay, and the last one is the Tarka, which is famous in the West Bengal. Okay, so now how we are going to move is that first of all we will be discussing about the etiology, then the host range, then the clinical science, then the habitat of the particular that bacteria, then the virulence and the pathogenicity, then the clinical science, then the diagnosis, then the prevention and control and all that. Okay, so moving on to the etiology of this particular disease, it is caused by Bacillus anthracis. Okay, it is a gram-positive bacteria which is non-motile and show catalase positive activity and oxidase negative activity. So this particular bacteria had two strains. Okay, it had two strains. One is the rough strain and next one is the smooth strain. So the rough strain of the Bacillus anthracis is virulent, while the smooth strain of the Bacillus anthracis is avirulent, right? So next peculiarity of this particular bacteria is that it do form the endospores. Okay, it likes the aerobic conditions. Okay, and it don't produce any capsule when it is grown in the laboratory conditions. Okay, now coming on to their host range. Okay, so the ruminants are highly susceptible for this particular disease. Okay, except there is an exception in the case of the ruminants, that the Algerian sheep is resistant towards what? Towards the anthrax disease. That's an important point that you have to notice. Okay, it is an important point. Now talking about the horses and the pigs, they are moderately susceptible. Okay, the carnivores are comparatively resistant for this particular disease, and the birds are totally resistant. It is also a point to important. Okay, it is also a point to notice over here. Now talking about the humans, as this disease is also zoonotic. Okay, anthrax is also zoonotic, guys. Okay, so it do affect the humans, and there are two types of situations that a human can be encountering. Okay, either it could be a cutaneous form of anthrax or a septicemic form of anthrax. So what happens in the cutaneous form? There is a formation of erythematous papules which are characterized by a black center, okay, and having a purplish tinge. Later on, in that particular condition of the cutaneous form, in the case of the humans, there is formation of Escher later. Okay, after the formation of the erythematous papule, then with the black center. It is the peculiarity of the malignant carbuncle that is caused by Bacillus anthracis in the case of the humans, right? The next form of the anthrax in the case of the humans is the septicemic form or the respiratory form, which is also known as wool sorter disease. Why it is called wool sorter? Because whenever the persons that are rearing sheep, they are going to what? They are going to do what? Sorting of the wools. And when they are going to do sorting of the wools, what happens that the spores of the anthrax is going to be inhaled by that particular person who is sorting the wools, which will lead to the what? Wool sorter disease. Okay, so that you have to be focused about. Okay, now talking about the route of the transmissions, ROT. Okay, so it could be either transmitted through the ingestion of the spore. Okay, either the person, what he is going to do is ingesting the spores, right? Either what he can do is the person can inhale the what? Your spores of the anthrax, right? Now the next way, it could be through contact, because it is, as I mentioned, that there are certain cutaneous lesion also in the case of the humans. So what happens when the person is in contact with any infected animal, or the leather or the fur or anything that has been derived from the animal source, whenever the person is in contact with that particular infected animal, it could lead to the cutaneous form of anthrax in the case of the humans, or in the case of the other animals through contact also. Okay, either it could be through feed, or also the spores also do sustain in the soil. We will discuss it later in the case of the habitat of the bacteria. Okay, now it could be transmitted mechanically also. Okay, the spores of the anthrax could be transmitted mechanically also by the blood sucking flies, right? Now talking about the habitat. Okay, So habitat of this particular bacteria is quite important because the alkaline soil, okay, so the alkaline soil favors the growth of that particular Bacillus anthracis, while the acidic soil reduces the chances of the growth of the Bacillus anthracis. Okay, so what happens, the area like Andhra Pradesh, okay, Andhra Pradesh, Tamil Nadu, Karnataka, okay, in India, these regions, what they have is alkaline soil, okay, and these regions having the alkaline soils have more cases of anthrax, right, as compared to the northern states of the India. Okay, because these states are having what, alkaline soil, which is going to favor the Bacillus anthracis, right. Now one more thing, that the humidity and temperature also supports the growth of the bacteria, right. We should be aware of that particular thing also. Okay, so the humidity and temperature do play a role in the habitat or the survivability of the particular bacteria, right. And this bacteria forms endospores and can survive more than 50 years in the soil. Okay, so one important thing to note over here that I will also discuss later on, but an important thing to discuss is, it is never recommended, not recommended to do PM if you're suspecting it to be anthrax. Okay, we do not perform postmortem if we are going to be suspecting that, oh, this particular case that we are going to visit, it could be anthrax. Okay, so we are not going to do postmortem on that particular animal. Why? Because whenever we are going to expose, whenever we are going to what we are doing is postmortem, we are going to open up the carcass. Okay, so when we are going to open up the carcass, what happens, the bacteria will be coming out. Okay, so consider this to be the body of animal, okay, that we had open. So this is the bacteria, right, this is the bacteria. So this bacteria is going to come in the environment and form spore, right. That spore can sustain over 50 years, right, and which is not good for us, good for the bacteria but not good for us, right. So it is not recommended to do postmortem whenever we are going to suspect that, oh, it could be the case of the anthrax, right. Please remember this, it is quite an important question also, right. Moving on to the virulence and the pathogenesis of the anthrax, right. So talking about, first of all, that there are 3 virulence factors, okay, we are going to discuss, and all those factors, all the 3 virulence factors do depends on the host temperature and the CO2 concentration inside the host. Okay, so talking about the very first virulence factor, which is the capsule, right. So this factor is plasmid encoded, which is designated as what, PXO2. Okay, it is composed of what, polygamma D-glutamic acid. Please remember this, okay, that the capsule of the anthrax is formed of what, that it is formed of polygamma D-glutamic acid, right. And what's the function of, because there is the virulence factor, what's the role of the virulence factor is to support the bacteria to cause the pathogenesis inside the host. That's what the role of the virulence factor is. Okay, now talking about the capsule, it is like what, it inhibits the phagocytosis and provides resistance towards the neutralizing antibodies that has been produced by the host. Okay, you should remember that what's the role of the virulence factor also. Okay, it is important. Now the next virulence factor is the your complex toxin. Now it is encoded by what, plasmid encoded only, and it is encoded by the PXO1 plasmid. Okay, now there are 3 components of the complex toxin, 3 antigenic components, right. 3 antigenic components of what, the complex toxin. So very first is the your protective antigen. Okay, remember that all three complex toxin antigenic determinants have what, their factor number and their short name. Okay, so the protective antigen is also designated as PA or factor number second. So what's the role of the protective antigen is, it provides the anti-phagocytic property, okay, and act as a binding moiety for the other 2 factor, which is the edema factor and the Leatham factor. What happens that this is the cell, This is the protective antigen which gives or pave a way for the other two factors for the host shell, right? So it is what it is doing, it is going to be acting as a binding moiety through which other two factor can act on the host cell and create a pathogenesis, okay, or create an harm to an animal, right? Now moving on to the other factor, which is the edema factor. Okay, so this edema factor is also designated as OF or as in factor number first. So okay, now what is the principal target for this edema factor? It is the neutrophil. Okay, so the principal target for the edema factor is the neutrophil. Now talking about this edema factor, it is what? It is a calmodulin dependent adenylate cyclase. Okay, so the edema factor is what? Calmodulin dependent adenylate cyclase. Okay, what happened? As I mentioned earlier, that also what is the function of the protective antigen? It acts as what? It acts as a binding moiety for the other two factors, right? Now what happens? This edema factor is going to bind on the your protective antigen. Okay, when it will be going to bind on the protective antigen, it will going to be entering the cell. Okay, it will increase the level of CAMP inside the cell. Okay, cyclic adenylate monophosphate. Okay, now it is going to increase the level of the CAMP inside the cells, right? So whenever there is an increase in the level of the CAMP, it will lead to what? It will lead to the disturbance of the water homeostasis, right? Okay, what happens that the level of the CAMP increased inside the cell due to the edema factor, it will lead to the disturbance in the water homeostasis, and when there will be disturbance in the water homeostasis or the imbalance in the electrolyte level inside the cells, it will lead to the accumulation of the fluid inside the cells, and it will lead to ultimately what? Edema, right? Now coming on to the next factor, which is the lethal factor. Okay, so what, same as earlier, that it also going to bind to the protective antigen. Okay, and then what it is doing, it is going to just before discussing that, it is what is this, it is designated as, it is designated as LF or the factor number third. Okay, it is what? It is a zinc metalloprotease. Okay, what was the edema factor was? It was the calmodulin dependent adenylate cyclase, while the lethal factor is what? Your zinc metalloprotease, right? Now what happened? It caused the death of the macrophages, the neutrophils, the epithelial cells, and the endothelial cells, and the dendritic cells, okay, which are the antigen presenting cells also. Okay, APC, these are the APCs, the dendritic cells. Okay, antigen presenting cells. Okay, now what it do is that it stimulates the macrophage to release the cytokines, the TNF alpha, the interleukin 1, and the free radicals, which will ultimately it will be leading to the shock and the death of the animal, because these all are against the response towards the bacteria, right? These all are released towards the response towards the bacteria, and it will ultimately lead to the systemic shock and the death of that particular animal. Okay, whenever there is an acute situation, it will ultimately lead to the death of the animal. If there will be large amount of spore ingestion or the inhalation by a particular animal, it will lead to the systemic shock and sudden death. Okay, there will be systemic shock in the chronic cases, or not the chronic cases, means the cases that are sustaining for a few more days, but the more sudden death would be in the case of the acute cases, in which the animal will be going to ingest a large amount of the spores of the anthrax, right? Now the last one is the spores. As I mentioned it earlier, the spores can enter through the skin or the mucous membrane and germinate at the site of the entry. Okay, I hope that particular pathogenesis and the virulencity is clear to you all, right? Moving towards the clinical signs. Okay, so what happens? Obviously, whenever a disease is there is pyrexia. Okay, in the case of the cattle and sheep, it causes a fatal condition, because as I had mentioned earlier, that Bacillus anthracis has a more susceptibility towards the ruminants. Okay, there would be congested mucosa. Petechiae may be observed in the antemortem of the particular animal. Okay, subcutaneous edema, as I had mentioned earlier, due to the edematous factor. Okay, there would be subcutaneous edema, we can see. Okay, now next few points are important. Okay, so there would be exudation of dark terry red color blood from the natural orifices, and what's the main thing is it is unclotted. Okay, this particular thing is what we can assure, we can't assure, okay, we can't assure that due to that particular unclotted blood that is coming out from the natural orifice, we can't assure that it is anthrax, because there are also scenarios in the other diseases where unclotted blood do come out. But what we can do is, yeah, we can just presume that, oh, it could be the case of anthrax, because in the case of the HCN poisoning also, there is no coagulation, no coagulation of the blood. Okay, so it could be an differential diagnosis case also. Okay, but we take a preventive measure before doing the PM examination, that we shouldn't open this carcass because it could be anthrax. Okay, that's why we do not open up the carcass whenever we see an unclotted blood that is coming out of the natural orifices of a particular animal. Okay, now coming on to the next point is that the splenic condition, there would be splenomegaly, and the appearance of the spleen will be like blackberry jam. Okay, there would be incomplete rigor mortis in the case of anthrax. Okay, and there is anthracoids. These are the postmortem invaders, most commonly seen in the case of the horses. The postmortem invaders, they are known as anthracoids, like Clostridium also. Okay, in the cases of the swine, there is an edematous swelling, as earlier, same thing, due to the edema factor, and the regional cervical lymphadenitis is also seen. There could be hemorrhagic enteritis, which will further lead to the dysentery. Okay, I hope this particular scenarios are clear to you. Okay, the clinical signs are quite important for the anthrax, especially the 6th, 7th, and the 8th one. Okay, now moving on towards the diagnosis. The diagnosis is also important. So what we are going to see is, like, whenever we are going to diagnose a particular disease, obviously the history and the clinical signs are the most important thing, which will help us to direct towards a particular diagnosis. Okay, so there may be a blotty condition that we can see. Now what we are going to do is that whenever we are going to form a culture for that particular suspected case, what happened when we are going to do what gelatin stab culture for that particular sample that we had acquired from that particular animal death site. Okay, it will lead to formation of what inverted fern tree like appearance. Okay, it is important to note. Okay, there are several points to important to note in the case of the diagnosis of an anthrax. Okay, there is a ground glass appearance which is having a flat colony, dryish grayish after the 48 hours of incubation. Okay, it is also important. Now about the colony, when we are discussing to about the colonies, there is formation of mucoid colonies, and it is grown on what, a media containing more than 5% of CO2. Okay, and having what, 0.7% of the bicarbonate. Means the anthrax, if the bacteria is anthrax, it is going to form mucoid colonies when it is grown on the media containing more than 5% of the CO2 concentration, and in addition there is what, 0.7% of the bicarbonate. Okay, now at lower magnification, okay, what we can see is medusa head colonies which are non-capsulated in aerobic condition in the blood agar. Okay, in the blood agar at lower magnification, what we can see is there is a formation of medusa head colonies. Okay, all the things that I had written in a separate pen, okay, those points are important. Okay, please note them out. Okay, now there is a dark terry unclotted blood from the natural orifices we had discussed. Now coming on to the McFadyen reaction, what we are going to do is we are forming a blood smear first. What we are going to do is, see, this is the blood, what, this is a slide. Okay, this is a slide. Okay, now we are going to take another slide, okay, and placing them at the angle of 45 degree. Okay, and now what we are going to do is the blood that we had collected from the site of the animal, okay, we are going to pour that blood, we have poured that blood on one end of the slide. Okay, now using the slide 2, this is the slide 2, this is slide 1. Okay, now using the slide 2, what we are going to do is we are going to spread this blood throughout the slide 1. Okay, now what happens, there will be formation of a thin blood smear. Now what we are going to do is we are going to place the mercury chloride as an fixation. Okay, we are going to for what, mercury chloride, sorry, okay, mercury chloride at the site, And what's the ratio it would be? It will be 1:1000, the concentration ratio for the mercury chloride. Okay, what is the function of the mercury chloride? It will fix this line. Okay, for 5 minutes we are going to pour it like that. Okay, now we are going to stain it with what? Polychrome methylene blue. Okay, when we are going to stain this particular slide with polychrome methylene blue, what happens, what we can see is there is blue stained rod. Okay, wait, there are blue stained, like this, blue stained rods. And what's the thing next is that it is surrounded by what? Pink capsule. That would be the appearance of that particular bacteria after staining from polychrome methylene blue. Okay, and what's the arrangement is? It's like the boxcar arrangement or a bamboo pole like appearance would be shown. Okay, after staining that particular bacteria. Now talking about what? Now we are going to talk about the pearl string reaction. It is also an important thing to discuss. The pearl string reaction is also important, right? What happens in the pearl string reaction is it is through the spot technique. Okay, it is through the spot technique. Okay, it is through the spot technique, and what we do is we inoculate the sample on the tryptosaccharide, which is containing what? This much amount of the penicillin. Okay, what happens? This is the culture plate, right? Having the tryptosaccharide. Okay, and this particular tryptosaccharide have what? 0.05 to 0.5 units of what? Penicillin, right? This is what having, right? The culture plate is having what? 0.05 to 0.5 units of the penicillin, right? Now what we are going to do is we are going to incubate it for 6 to 18 hours at a temperature of 37 degrees Celsius. What we are going to observe is that there will be formation of string or the pearl-like appearance. Okay, this is all about the pearl string reaction. All the diagnostic procedures for the anthrax are important, please note that. Okay, now moving towards the Ascoli test. It is a thermoprecipitation test to detect the antigen of the Bacillus anthracis in the biological material, which we take what? We take a part of the muscle. Okay, the muzzle of the animal. We take a part of the muscle of the animal. Okay, then we are going to perform what? A thermoprecipitation test. Furthermore, for the anthrax, we can do what? We can do the AGID, okay, agar gel immunodiffusion test, or the CFT, okay, or the ELISA. Okay, this was all about the diagnosis. Now talking about the control measures. So there is a vaccine which is known as the spore vaccine, and what happens that as a preventive measure, what you should do that if this is your farm, you should always have a foot bath. Okay, foot bath at entrance. Okay, at entrance point there should be a measure having a foot bath. Okay, such that there is no further contamination or the bacteria that would be entering inside your farm. Okay, there should be foot bath procedure of the foot bath in your farm having the 5% formalin. If there is an case that you had encounter in your farm which is suspected to be anthrax, so a proper fumigation should be done at your farm. Okay, and as I mentioned earlier, do not open the carcass for the postmortem examination if you are going to suspect that it could be anthrax. Okay, and then at last, bury the carcass 6 feet below with a quicklime, or either you can incinerate or burn it down. Okay, so that was all about the anthrax. Let me show you certain pictures of also anthrax. Yeah, here they are. Okay, so here you can see there is unclotted blood coming out from the natural orifice, which is the ear or the nostrils or the mouth. Okay, you can clearly see the blood is coming out from the rectum also. Okay, so this blood is unclotted. So in these cases where we can see that the unclotted blood is oozing out from the natural orifices, we can suspect it to be anthrax, but we cannot assure that it can be anthrax, because there are many diseases or many poisoning conditions, okay, in which the similar condition could be observed. Okay, but it is better, do not open the carcass if you're going to see cases like this. You can also see here also that the blood is oozing out from the eyes also. Okay, so this was all about anthrax. Okay, thank you everyone.